Uh, National Medical Commissions last year had made it mandatory for all the medical colleges last year in the month of October to have an antimicrobial stewardship program mandatory in all the medical colleges. While it is also included in the chapter 5 of NABH standards to have an antimicrobial stewardship program. The chapter 5 hospital infection control of NABH states that the organization has an effective antimicrobial management program through regularly updated antibiotic policy based on local data and monitors its implementation. The program also includes monitoring of antimicrobial usage in the organization. To explain in short, the organization should have an antimicrobial stewardship program with an updated antibiotic policy, which needs to be prepared based on the local antibiogram and needs to be reviewed regularly. And also it should include monitoring of the same. Now, what is antimicrobial stewardship? Antimicrobial stewardship and antimicrobial stewardship is can be explained as in terms of 7 R's. That is right drug against the right bug, that's the right antibiotic against the right pathogen for the right person at the right time, at the right dose, right duration, right frequency, and through the right route. In short, it's the rational antimicrobial usage that constitutes our antimicrobial stewardship. One important component of this antimicrobial stewardship is the antibiotic policy, or preferred term will be an antimicrobial policy, which means the right drug for the right bug. Now, what is the aim of this antibiotic policy? The first most aim is to select the patients who needs to be treated. We often see the patients who are colonized or patients whose clinical sample is contaminant are often falsely treated, or they are antibiotics are higher antibiotics are prescribed when not needed, or antibiotics are prescribed when there are viral infection and many other causes. So the first aim of the antibiotic policy is to select the patients who really needs to be treated. Second, to have all the antibiotics available in the formulary. So the second aim of the policy is to have, we should know what the antibiotic that should be available in our healthcare setting for the prophylaxis or the treatment of the patient. Third aim is to avoid unnecessary antimicrobial usage. So this constitutes the aim of the antibiotic policy, that is to select patients, then to have all the antibiotics available in the formulary and avoid unnecessary antibiotic use. Now, what is the base of this base of the antibiotic policy? Spectrum. What is needed to constitute the antibiotic policy? We should know the spectrum of the antibiotic activity. While preparing an antibiotic policy, we should know the spectrum, whether for a particular infection, whether we need a narrow spectrum or a broad spectrum. There are narrow spectrum antibiotics like clindamycin, metronidazole, and others. There are broad spectrum antibiotics like macrolides and others. So the spectrum of antimicrobial activity needs to be under consideration while preparing antibiotic policy. Then we should know the pharmacokinetics and pharmacodynamics. Pharmacokinetics, that is the absorption, distribution, metabolism, excretion of the antimicrobials, needs to be considered while preparing antimicrobial policy. As also the pharmacodynamics of the antimicrobials, that is the site of the action, the mechanism of action, the mechanism of resistance against that antimicrobials needs to be considered while we are preparing an antimicrobial policy. Similarly, the adverse effects, the complications, the drug-to-drug interactions needs to be considered for preparing policy. Also, the most important, the potential to select resistance. Some of the antimicrobials such as colistin, polymyxin, tetracycline have the potential for selective resistance, and they need to be reserved. So that's such the protocol for the reserved or the restricted antimicrobials needs to be there in our policy. Second, cost effectiveness. Then cost effectiveness of the antimicrobials needs to be considered while preparing antimicrobial policy, and special needs to be given, special attention to be given while preparing antibiotic policy for individual patient groups, like for immunocompromised patients or for dialysis patients or chemotherapy patients and other such group of patients. So this forms the basis of the antibiotic policy. What are the key elements of this policy? The antibody policy is made by is prepared by multidisciplinary group. As you can see from this image, it's a the departmental heads, the HODs of the departments of the clinical departments should play a key role along with the other disciplinary healthcare personnel like microbiologists or the pharmacists or the intensivists or the eye physicians. Along with this team, the HODs of the department also should be involved. It is a multidisciplinary, activity to prepare an antibiotic policy. Second, the antibiotic policy should be adapted from pre-existing evidence-based guidelines to suit the local needs. Now we have a national antimicrobial policy and the national antimicrobial resistance data also. So based on this antimicrobial resistance data and based on this national antimicrobial policy as a template, where the healthcare, a particular healthcare setting should prepare its own antibiotic policy. Then the policy should be based on systematic review of the local AST, that is, and surveillance data. So every healthcare setting should have their own antibiogram. Antibiogram, that is the presentation of the susceptible pattern of the pathogens prevalent in their healthcare setting. So every healthcare setting should know which are the predominant organisms present in their healthcare setting and against which antibiotics they are sensitive and resistant, and the frequency of the same. So based on this data, the antimicrobial policy should be prepared for a particular healthcare setting. Then recommended antibiotics should be effective against the pathogens of that locality. I said before, the antibiotic, whatever has been stated in the antibiotic policy, should be active against the local pathogens. A single formula, single antibiotic policy of one hospital cannot be applied to other hospital of another area. So it differs from hospital to hospital and from state to state and others, because the local antibiogram of every healthcare setting differs. Then in the antimicrobial policy, there needs to be mention of the recommended dosage, the route and duration must be mentioned. Then identify the evidence that is lacking. When you prepare an antibiogram and for an antibiotic policy, see what is the evidence that is lacking, and research should be made to fill up those lacuna wherever we are lacking, wherever the knowledge is insufficient or not satisfactory. The research should be carried on to fulfill those requirements. Then auditing of the antibiogram or the antibiotic policy or the reviewing of this antibiotic policy should be done at regular intervals. Based on the changing antibiogram every 6 monthly or at least annual basis, the antibiotic policy should be reviewed accordingly. And the policy should be reviewed by experts who are not part of the developing group. So the developing group members should not be involved in reviewing of the policy. Then the next slide is the antibiotic sensitivity overview. While preparing the antibiotic policy, as I said before, as the basis of the antibiotic policy, the spectrum of the antibiotic action should be under consideration. For example, as you can see from this slide, penicillin is the antibiotic which is active against streptococci, or amoxicillin is active against streptococci, E. coli, and Klebsiella. Similarly, clindamycin against anaerobes, or clindamycin against MRSA, MSSA, streptococci, or vancomycin, ticopan, linezolid, and daptomycin against gram-positive cocci, but no action against gram-negative bacilli. Similarly, ciproflox against a wide range of gram-positive cocci and gram-negative bacilli. So similar, the mechanism, the spectrum of this antimicrobial sensitivity should be considered while preparing the antimicrobial policy. The most important is know your local antibiogram. The antibiogram is the basis, is the driver for preparing the antibiotic policy for the implementation of the stewardship strategies. To prepare an antibiogram, the Clinical and Laboratory Standard Institute, CLSI M39A4 document. CLSI stands for Clinical Laboratory Standard Institute. It has given the guidelines for the preparation for the analysis and presentation of cumulative antimicrobial susceptibility test data. So you can refer to the healthcare setting, can refer to the CLSI M39A4 document for the analysis and presentation of their antimicrobial susceptibility test data, and accordingly prepare their antibiogram. Now, what is the CLSI recommendations recommends for antibiogram preparation? Analyze or present the antibiogram at least annually. So on an annual basis, or if the antimicrobial susceptibility data is sufficient, you can prepare it or analyze it every 6 monthly. include only the final verified test results. The provisional test results or the should be avoided, and only the final verified test results should be taken for preparing antibiogram. Then more than or equal to 30 isolates should be examined for preparing antibiogram. Then environmental surveillance isolates, such as those organisms which have been isolated from the OT samples, surface swabbing or air sampling, they should not be considered while preparing the antibiogram of the healthcare setting. Then eliminate the duplicates. Only when a patient is infected, say the bloodstream infection, he can grow the Klebsiella at a number of times during his stay in the hospital. So only the first time when the isolate was identified, that the first isolate who the sensitivity of the first isolate should be taken into consideration while preparing the antibiogram. Then only those drugs who are routinely tested and reported. It will become very complex if we prepare the antibiogram taking into consideration all the antimicrobials of our formulary. So one of the few commonly prescribed or routinely tested drugs needs to be considered while preparing antimicrobial policy. And one is the susceptible and not the intermediate. One is the susceptible antibiotics should be considered for policy preparation. So we have a WhoNet. It's most of the healthcare settings. It is a manual based antibiogram preparation, but there is a software called WhoNet. It is an automated electronic health record based antibiogram. It's a software which can be used for making antibiograms. It is freely available on the internet, and we can make use of this WhoNet. To make an antibiogram. It is an EHR, that is electronic health record based antibiogram. The particular microbiology technician or this lab personnel enters the data of the susceptibility pattern of a particular pathogen against the different antibiotics into this WhoNet software, and on a regular basis we get the antibiogram prepared from this WhoNet. If the healthcare setting has automated platforms like Vitech or Phoenix, these automated platforms like Vitech has a Myla software, or the Phoenix has an epicenter. This software too provides a real-time antibiogram, say sample-wise, pathogen-wise, and department-wise. We can get an antibiogram. That is the presentation of the susceptibility of different pathogens against different antibiotics on a real-time basis. So that's the advantage of this software of this automated platform. Otherwise, we will discuss in ahead how to prepare manually the antibiogram, or else we can make use of the WhoNet software, which is freely available for preparing the antibiograms. Notice what is let's see the overview of a cumulative antibiogram. In a cumulative antibiogram, we get a statistical significance of changes in the percentage of susceptible isolates. When we prepare an antibiogram, in a cumulative antibiogram, we can get the percentage of the susceptible isolates, so that we can accordingly make the interventions in our antimicrobial stewardship program. Then the antibiogram should include species with more than 30 isolates, as said before. Include the first isolate of the patients, not the duplicate isolates or subsequently isolated isolates, and then certify the percentage for the multi-drug resistant pathogen organism. So focus should be there on the multi-drug resistant pathogens like Pseudomonas, Acinetobacter, or MRIs and others while preparing the antibiogram. Compare the local and the national antibiogram. See whether we are drastically different or we are closer to the national antibiogram, and accordingly test and prepare our antibiogram for further making the antibiotic policy. And compile and report the antibiogram and it trained annually. Changing the empirical therapy based on this antibiogram should be done on a regular basis. Now let's see how to prepare the antibiogram. I am focusing more on the antibiogram because on the basis of the antibiogram we will be preparing our antibiotic policy. So the first thing is compile the local hospital data sensitivity pattern based on the site of infection. Geographical we can prepare antibiogram for different geographical sites like ICUs or wards or surgical ward or surgical ICU or and cath lab and dialysis areas and others. So here we can have a frequency of the organisms identified from the different geographical areas. Say from ward area we can prepare a chart or a pie chart wherein we can get the frequency of the different organisms. As you can see from this image, we from this pie chart, we get to know that from a particular general ward, the Klebsiella is more prominent, followed by E. coli. E. coli is prominent, followed by Klebsiella or Pseudomonas. So this when preparing antibiogram, so pie charts will help us, will help the clinician or the drug and therapeutic committee to easily identify the predominant organisms of a particular department of the healthcare setting. Say for example, from this general ward, E. coli, Klebsiella, and Pseudomonas are more common. Then we can have the resistance profile. So the sensitivity pattern of different antibiotics or antimicrobials against the different pathogens. Say we can have the sensitivity patterns of the Enterobacteria. And can be of different from different areas like ward or ISOs and others. And put this data, the frequency of the organisms identified and the sensitivity pattern in a given template. Now, what is the template? We'll see here. This template should also include the site of infection and the type of infection, the causative pathogens, and preferably a 6-month data or more than or equal to 30 isolates should be considered. And capture the pathogens, not we can, as like antimicrobials, not all antimicrobials we can consider. Similarly, capture one of those pathogens which contribute to about 80 to 90% of the infections. So one of those commonly isolated pathogens should be considered while preparing antimicrobial policy. And capture the susceptibility from the highest susceptibility to the lowest susceptibility while preparing the policy for particular syndromes like pneumonia or UTI or bloodstream infection or surgical infection and others. For example, use this friendly toolkit to put the data based on the site of infection. Toolkit will be separate, can be separate for ward and the ICU isolates. Toolkit should consist of the 5 most common pathogens and most antibiotics in the decreasing order of the sensitivity. So we can select the 5 most common antibiotics, and from the higher susceptibility to lower susceptibility, we can plot out their surveillance data. The toolkit should also contain the validity period. Let's see from an example. Identify the 5 most common pathogens. The 5 most common pathogens can be Pseudomonas, Klebsiella, Acinetobacter, E. coli, Staph. Then the next step is to find out the susceptibility, the 5 most highest susceptibility, lowest susceptibility, the 5 most sensitive antibiotics against each of these 5 isolated organisms. Say for Pseudomonas, peptase, imipenem, ertapenem, cephalosporin, salpetem, cetirizine. Similarly for Klebsiella, peptase, imipenem. So 5 the commonest susceptible antibiotics should be plotted against the 5 commonest organisms to prepare an antibiogram. So this is how our antibiogram will look if we are preparing it on a manual basis. The 5 most common organisms, the Pseudomonas, Klebsiella, Acinetobacter, E. coli, and Staph aureus, the prevalence is plotted in the second column, and against each of the organisms, then we have the 5 most susceptible antibiotics. For example, Pseudomonas, the prevalence is 30%, and the 5 common and susceptible antibiotics are colistin, imipenem, cephalosporin, sulbactam, peptase, amikacin. Similarly, we can chart out other common susceptible antibiotics with their against our pathogen with their frequency. So this is how the manually as an antibiogram can be prepared. So this is similar. The antibiogram can be prepared against bloodstream infection, urinary tract infection, or surgical infection, pneumonia for a particular setting like ward or ICU. This is how a sample antibiogram looks like against the particular organisms of a particular areas against different antibiotics. The sample antibiogram consists of the sensitivity percentage. Stratified, we can have a stratified antibiogram based on age wise, whether we can have an antibiogram for pediatric group separate and for adult group separate. We can have an antibiogram separate for syndrome wise, for bloodstream infection or urinary tract infection, and we can have an IPD versus OPD antibiogram similarly. While preparing antimicrobial policy, also we need to take into consideration the intrinsic resistance or natural resistance. Some of the pathogens some of the pathogens bear a natural resistance, means or intrinsic resistance, means these are pathogens, they are natural resistant to some of the antibiotics because due to some chromosomal inherited resistance mechanism. For example, Klebsiella is natural resistant to ampicillin or amoxicillin. Pseudomonas is natural resistant to tigecycline, cotrimazole, and ampicillin. Similarly, all gram-negatives are natural resistant to vancomycin. All aerobic bacteria are natural resistant to metronidazole. So this natural resistance pattern should be taken into consideration while preparing antimicrobial policy. Now, patient while preparing antibiotic policy, we cannot make an antibiotic applicable to all the patients. The patient needs to be stratified. Now, why the patient risk stratification is needed? Not all the patients admitted in a hospital needs the broader spectrum antibiotics. So the risk stratification is necessary to decide who needs the narrow spectrum and who needs the broad spectrum and who needs the broadest spectrum. Now, how for risk stratification of the patients, three factors are taken into consideration. One is contact with the healthcare system, whether the patient has a contact with, whether he has no contact or contact with the healthcare, or that is recent admission or nursing without invasive procedures, or long hospitalization with invasive procedures. So contact with the healthcare system is taken into consideration while risk stratification of the patients. Then second is antibiotic treatment, whether the patient was on treatment or not, or a recent treatment with antibiotics. Then thirdly, patient characteristics, like whether the patient is young with comorbid condition, whether he's old or with multiple comorbidities, or whether the patient has cystic fibrosis, lung disease, and other such immune deficiency disorders in the patient. So these three factors need to be considered while risk stratification of the patients. The risk stratification of the patient can be done into 4 groups, considering these 3 factors. Patient type 1. Now, for each of these groups of the patients, we can have we have an antimicrobial policy accordingly for these 4 groups of patients. The type 1 group of patients includes no contact with the healthcare system or no prior antibiotic treatment in the last 90 days, and the patient is young with few comorbid conditions. In this group of patients, their bacterial infections include with minimal risk of multi-drug resistant pathogens like ESBL producing Enterobacteria or MRSA, or non-fermenters like Pseudomonas and Acinetobacter. So this group of pathogens are very less predominant in this group of patients, and invasive fungal infections are unlikely in this group of patients. While in the patient type 2, there is a contact with the healthcare system. Antibiotic therapy has been given the last 90 days. The patient can be old with more than 65 years with multiple comorbidities. In this group of patients, there is a risk of bacterial infection with pathogens like ESBL producing Enterobacteria and MRSA, or there is a minimal risk of non-fermenters like Pseudomonas and Acinetobacter, and there is a minimal risk of invasive fungal infections. As we compare with the patient 1 and 2 with the type 3 patient group, here the patient have an hospitalization of more than 5 days and or infection following invasive procedures. They have recent or multiple antibiotic therapies. The patient characteristics include cystic fibrosis or lung disease or advanced states, neutropenia, and other such immunodeficiency disorders. And there is a high risk of bacterial infections with such as multi-drug resistant pathogens like ESBL producing Enterobacteria or MRSA, and non-fermenters like Pseudomonas and Acinetobacter. The risk of invasive fungal infection is also high in this group of patients, particularly in special cases like patient undergoing transplant or chemotherapy. Patient type 4 is the most serious group of patient group. Here in the hospital is more than 5 days or ICU stay of more than 3 days, and there is no response to the broad spectrum antibiotic after 96 hours of treatment. And there are additional risk factors like hemodialysis or surgery and others. In this group of patients, the risk of bacterial infection is the highest, especially with multi-drug resistant pathogens like Pseudomonas, Burkholderia, Acinetobacter, and others. And there is a high risk of fungal infections. So this was about that. This was about the 4 group of patients, and we'll be preparing the antimicrobial policy accordingly to each separate for each of these 4 group of patients. This policy can be either empirical or prophylactic or therapeutic. Empirical antibiotic policy means we know that there is a patient with an infection, but we are not aware of the culture and sensitivity test result of that of the particular infection sample. Prophylactic, to prevent infections, we can have an antimicrobial policy. Therapeutic, when we are sure of a patient with an infection and the culture and sensitive results are available. So what is the principle of empirical antibiotic therapy? Let us see. Let us see to each of these principles of empirical therapy. When we think of an empirical therapy, 4 factors need to be considered. Is the patient infectious? Which organ is sick? Then third, which antibiotics should be used and what dose? The first thing, is the patient infectious? Is the patient infectious? For this, urgent antimicrobial therapy is warranted when the patient is in sepsis. Every hour delay in the sepsis, every hour delay for an antimicrobial treatment in sepsis increases the mortality chances by 3%. So antimicrobial therapy should be initiated within 1 hour of recognition of sepsis. So patient infectiousness needs to be considered while empirical therapy. then get appropriate cultures and start the therapy. Identification of the infective organisms can be done from rapid method like gram stain, especially in sterile fluid like CSF, pleural fluid, and others. Then also we can get an idea of the infective status from the buffy coat of the blood, which reveals organisms within polymorphs. From that, we can get to know whether the patient is highly infectious. Then immunological methods like antigen detection can help us to identify patients with infections. Then molecular detection of the pathogens like we have biofire and other such a microarray panel from which we can identify the pathogens on an immediate basis. Now similarly, we have MALDI-TOF, that is matrix assisted laser disruption ionization time of flight. That is MALDI-TOF, which identifies the organism within seconds from the culture colonies. So such molecular methods can be used for identifying the infection from the patient sample. But the gold standard method is the culture. And when we do a culture, ideally the culture should be samples should be sent for culture before starting antimicrobial therapy. Even if the antimicrobial therapy has already been initiated, then after the first half-life of the antimicrobial, or before starting the second dose of the antimicrobial, this sample should be sent for culture under aseptic precautions. If the patient is in sepsis and blood culture needs to be sent, then ideally, as per recommendations, as per guidelines, 2 sets of blood culture should be taken for culture. Now, what does two sets means? One set means 1 prick. That is 1 prick from a left hand should consist of an aerobic and an anaerobic bottle, and the second set should be from the right hand. That is 1 aerobic and 1 anaerobic bottle from the second prick of the right hand. If the patient is on a central line, one similarly aerobic and anaerobic set should be bottles should be collected from the central line. Now, why so much emphasis on these paired bottles? Not all the organisms are strictly aerobic. Not all the organisms are strictly anaerobic. Most of the organisms, 70 to 80% of the organisms are facultative anaerobes, like E. coli, Klebsiella, Staph. They are facultative anaerobes. That is, they can grow in the presence or absence of oxygen. So it has been observed that these pathogens get isolated 4 to 6 hours earlier than from the anaerobic bottles than the aerobic bottles. It is the anaerobic bottles which flags first 3 to 4 hours earlier than the aerobic bottles. Also, most of the times aerobic bottle doesn't flag and anaerobic bottles flag. So hence the necessity to have a paired bottle, that is aerobic and anaerobic bottle from both the hands. The second advantage is not only we get the yield of the pathogens from the paired bottles, but also it helps us to rule out the colonization or the contamination. Then then the next thing, we need to remember the pathogens causing infection in various sites. We need to be aware of the commensals or the colonizers of the different sites of the body. So the spectrum of the antimicrobial needs to be considered. That is broad spectrum or narrow spectrum, and accordingly, escalation or de-escalation should be done based on the culture results. When we identify a patient with infection with an antimicrobial therapy, the antimicrobial needs to be considered for tissue penetration. It is important to choose those antibiotics that penetrate into the organ concerned. For example, it's a meningitis, then ceftriaxone or the cefotaxone should be considered. If it is an urinary tract infection, then tigecycline won't be an effective. Then for such infection, if it's a nitrofurantoin or norflux or fosfomycin should be considered. So this tissue penetration on the site, the antimicrobial should be chosen such that it is active at the site of the infection. Then the resistance pattern, the intrinsic resistance or the local antibiogram resistance pattern should be taken into consideration while empirical therapy. The host factors like age and other such comorbid factors need to be considered. So we have 4 group of patients. When the patient type 1, there should be limited use of broad spectrum antimicrobials. Here we have seen there is no contact with the health, and there is a health system, and that fungal infections are rare. So there is no role of an antifungal agents in type 1 group. In the type 2, there is lesser chance of ES belly infections, so ES belly infections to be treated if suspected with non-pseudomonal antibiotics like group 1 carbapenems, like imipenem or beta-lactam inhibitors can also be preferred for mild ES belly infections. Vancomycin or teicoplanin can be considered for MRSA. Here in this patient type 2 also, there is no role of an antifungal agents. In type 3, prophylaxis for fungal infection can be given as per IDSA guideline, as the Infectious Disease Society America guidelines. Here, the bacterial infection should be treated with broad spectrum antibiotics like group 2 carbapenems or anti-pseudomonal beta-lactam inhibitors in combination with fluoroquinolones or aminoglycosides or glycopeptides. In type 4, empirical therapy of fungal. Now, in type 3, there was a prophylactic fungal therapy. In type 4, there is an empirical treatment of fungal infections. And here in bacterial infections should be treated with a combination of antibacterials suggested for pan-resistant bacteria such as Pseudomonas burkholderia using alternate drug delivery systems or based on PKPD parameters, that is pharmacokinetics, pharmacodynamics. So the type, to summarize, all the type 4 patient group antibiotic choice differs. We have 3 major group of antibiotics. One is the access group, second is the watch group, and third is the reserve group. In the access group, this group of antibiotics includes antibiotics that have activity against a wide range of commonly encountered pathogens and a lesser potential for resistance. And this includes antimicrobials like amikacin, amoxicillin, ampicillin, amoxicillin, doxycycline, and others. Watch group are those group of antibiotics which has the potential for resistance, but we don't restrict it. We prescribe it under careful consideration, considering the resistant potential. And this group of antibiotics includes azithromycin, cefotaxime, ceftazidime, and others. Reserve group of the antibiotics are the most needs to be prioritized. They should be prescribed only after consultation with an ID physician or a senior consultant who has knowledge of infectious diseases. And this reserve group includes antibiotics like ceftazidime, ibactam, fosfomycin, colistin, polymyxin B, and others. So when you're preparing the antimicrobial policy, a reserved antibiotic policy needs to be in place to prevent for selective prescription of antimicrobials of those reserved group of antibiotics with the specific indications. So this is an example of an antibiotic policy for a bloodstream infection. Chart out the most common pathogens of the healthcare setting, say it is the antibiotic and others. Then chart out the antimicrobial therapy for each of the 4 group of patients, type 1 to type 2 to type 3 and type 4. But indicate presumptive therapy or the prophylactic therapy. Indicate that after culture report whether there will be escalation, what will be the criteria for the escalation, and what will be the criteria for de-escalation. Similarly, consider the indication of when to do the IV to oral switch, when to do the optimal, how to do a dose optimization. So all these factors needs to be put in place in while preparing an antimicrobial policy for syndrome wise. Now, when we give prophylactic antibiotics, say for surgical site infection, wherein the most prophylactic antibiotics are given, it should be given within 1 hour of incision. There is no point in closing the door after the teeth have escaped. So the more the chances, the more we are more hours apart from the incision time, there is the more the chances of infection. As you can see from this chart, if the prophylactic antibiotics, say for CABG, cefazolin or cefazolin has been given 3 to 4 hours earlier than this incision time, the chances of infection is more. If you give it to prolong after the after the incision time, against this chances of surgical site infection is more. So it should be given at the time of incision or within 1 hour of incision. The protocol differs for aminoglycoside. For aminoglycoside, it is 2 hours from the site, so from the time of incision. So antibiotics will be given 1 hour before the proceeding with the incision or at the time of induction anesthesia. Or second dose may be given if the surgery prolongs more than 3 hours. So if the surgery is more than 3 hours, the second dose may be given, except for vancomycin, which is a once a day prophylactic drug, or aminoglycosides and fluoroquinolones. We all know that vancomycin, aminoglycoside, and fluoroquinolones are time-based antimicrobials. That is, they need a once a day antimicrobial prescription. So for such antimicrobial, the second dose of the prophylactic antimicrobial should not be given. Prophylaxis antibiotics should be discontinued within 24 hours of the end of surgery, except for CABG, where it can be given till 48 hours. Otherwise, the antibiotics should be discontinued. See, when a surgery takes place, immediately if fibrin clot develops at the site of the surgical site of the site of surgery. So there is no point in giving the antimicrobial once after 3 hours or after 24 hours after the fibrin clot is developed at the surgical site, because the antimicrobial won't get an entry into the surgical site, and unnecessarily there will be an adverse effect or drug resistance. So we can we have seen the the different guidelines for antimicrobial policy, how to make an antibiogram. Similarly, we can make use of this template from treatment guidelines for antimicrobial usage in common symptoms, which is given by ICMR, or we can take a help of WHO guidelines, which gives a step-by-step approach for implementation of antibiotic policy and the guidelines. Now, when to say no to the higher antibiotics or de-escalation? What is the criteria for de-escalation or stop orders? It is a culture-based de-escalation. Whenever there is a suspected infection, we give an initial treatment. It should be a wide spectrum, but it needs to be immediately de-escalated once the lab confirmed culture and sensitivity reports gets available. So culture, it has been observed while I do an assessment of hospitals for antimicrobial stewardship. Often it is a clinical based escalation and de-escalation. Often the culture and sensitivity reports are often skipped. So it should not be that. So the culture and sensitivity should be done for the escalation and de-escalation of the patient treatment. And then there should be criteria in the antimicrobial policy when to switch from IV to oral. IV to oral switch is an important one of the important antimicrobial stewardship strategy. When the patient is able to take oral feeding, or the patient is stable and his vital parameters has been stabilized and normal, then the patient can be taken from IV to oral. So the protocol, that policy should be in place in the antibiotic policy. So what constitutes our antibiotic care bundles? The site of infection, the risk stratification, the local antibiogram, and the de-escalation strategies. This all combined for makes the education and an evidence-based antibiotic care bundle. Now, creating the therapeutic antibiotic guidelines. Choose one infection at a time. Look for the epidemiology of the last 6 months. Develop the antibiotic guidelines, including the local epidemiology. Do a consensus with these MDs or the specialists that work in the area. Listen to the doubts, ask about difficulties, and once the consensus is agreed, it should be implemented and followed up with the MDs or the specialists with the specific patients, and feedback should be taken for the same. Now, when we select an antimicrobial for a particular infection, the anti-selection should be very appropriate based on the susceptibility and susceptible test result and timing. The particular antimicrobial against the particular infection should be adequate. As said before, it should be able to penetrate at the site of the infection. Third, it should be optimal. It should be the antibiotic selected should satisfy the pharmacokinetics and the pharmacodynamic parameters. As we all know, some of the antibiotics like penicillin, cephalosporins, carbapenems are time-based antibiotics. They need to be given at regular intervals. While antibiotics like aminoglycosides and others are concentration-based antibiotics, they need once a day daily dosing. While some of the antibiotics like quinolones are area under concentration antibiotics. So there is a different group of antibiotics whose the PKPD differs. So that PKPD parameters needs to be considered while selecting the antimicrobial for a particular infection. To give an example, when we give a culture and sensitivity, when we give it a culture and sensitivity result, there are different types of antibiotics which are sensitive and resistant. Now, which antibiotics should be taken for selection? When there are 4 rules. First rule is always start with the beta-lactam if possible, especially in severe infection, because the beta-lactam have the best data supporting their use and are in general excellent drugs, except atypical infections. The second rule, do not compare the MICs between drugs. As said before, every antimicrobial has different pharmacokinetics and pharmacodynamics. That is different serum and tissue concentration and different types of action, like time different, concentration different. So do not compare the MICs. The MICs of different antimicrobials against different pathogens differs. If you are, when you see an antimicrobial susceptibility result, if you see less than or equal to, immediately don't go for the selection of the particular antimicrobial. Always also check the site of action of the antimicrobial, whether it achieves the pharmacokinetics or pharmacodynamics, whether the antimicrobial has an inducible resistance for that particular pathogen. For example, enterococci, they have interesting resistance to aminoglycosides, cephalosporins, cotrimoxazoles, and salicylates. So such which is the commonest isolated pathogen and the commonly which is resistant to commonly prescribed antibiotics. So natural resistance needs to be taken into consideration while selecting antibiotic. The patient-specific factors as said before, and the drug cost. Next thing, microbiology has always more information than what is put on the test report. So if more information is needed, the microbiologist can be approached, or the lab, we can get an extra information from the lab. We'll see an example ahead. Now, see this example from the blood culture. Enterococcus has been isolated, which is sensitive to ampicillin, dapto, penicillin, vancomycin. MSCs are well under the sensitive range. Now, which antibiotics should be tested? Should be selected. Now, first rule is always start with a beta-lactam if possible. Now, now the drug of choice will be, hence in this case will be ampicillin, because it is ampicillin sensitive. The second rule is do not compare the MSCs. Daptomycin is not better than ampicillin because the MSC is lower. Hence, ampicillin is still the drug of choice if sensitive. Also, if microbiology has more data, so we can also test for the linezolid also, but which is an oral option. But because linezolid is not cost effective, ampicillin is still preferred. Similarly, daptomycin, though sensitive, is not cost effective, ampicillin will be preferred over vancomycin and daptomycin. So, drug of choice will be ampicillin for this particular case. If we take another example of from Klebsiella isolated from urine culture, which is sensitive to which is ESB producing but sensitive to cefotetan and sensitive to meropenem, sensitive to levofloxacin but resistant to ciprofloxacin, sensitive to nitrofurantoin and sensitive to tigecycline. It is an urinary tract infection. Now we need more information whether it is cystitis or pyelonephritis. If cystitis, whether it's a male or female, if female, how old she is. Let's assume this is a cystitis in a young adult female with no more comorbid condition. Then you should think whether you want an IV route or oral route. Now, regarding the selection of antimicrobial, consider for this particular infection, urine culture with Klebsiella, it's an ESB producing. So even cefotetan was sensitive and meropenem was sensitive, we will not give cefotetan because it's an ESB producing organism. Also, if it is there was a less than, if you see the cefotetan will not be used because in ESB producing, meropenem is appropriate, but meropenem is IV. So the meropenem will also not be considered, will be less preferred. Ciprofloxacin is resistant, so levofloxacin will not also be used because ciprofloxacin is resistant. Tigecycline is sensitive, but tigecycline has a poor urine penetration. Hence, we are left only with nitrofurantoin. So nitrofurantoin will be the drug of choice. Again, microbiology has more information than what is reported. So we can, the clinician can approach the microbiology lab for the phosphomycin, which can be tested, which is not reported here. But my phosphomycin can be expensive, hence our drug of choice will be nitrofurantoin. So that was how about in my presentation about the antibiogram, the risk stratification of patients, what are the factors need to be considered for preparing antibiotic policy, and how to select a particular antimicrobial. To summarize, in any case, the results of sensitive testing need to be interpreted with some knowledge of biology of the organism and patient history before treatment is prescribed. Do not use the antimicrobials if they are not necessary. Always take into account the natural resistance pattern. Check for any unusual result. The patient history is very important. Drug choices need to be taken into account. The pharmacokinetics and the pharmacodynamics, dosage adjustment should be done. Optimization should be done to our sublethal concentration, and always possible use a narrow spectrum and resort to most important antibiotics like choline, tigecycline, and others, except in certain cases. So that was about in today's my presentation. Open for questions.
Most of us think that the set of genetic codes that we are endowed with when we are born dictates our destiny. So epigenetics is the science which elucidates very clearly why our genes are not our destiny. So in my topic, I would first be covering, giving you a brief about what is epigenetics and what are the different modalities of epigenetic modification which occurs. So one important one which has a lot of clinical usage is DNA methylation. I would be covering histone modification. How do we do it in the labs? A little bit of data, and then cover the diagnostic tests. Just one slide on the diagnostic tests and what are the epigenetic therapeutic drugs which are presently available. So the world first got sensitized about this topic of epigenetics when there was this article which was published in Time, which was known as Why Your DNA Isn't Your Destiny. And this was published by Mr. John Cloud, who happened to be covering health for Time. So this study was basically done by Dr. Lo Bygren. The study is the one which was published in Time. And Dr. Lo Bygren happened to be a public health specialist who was working at the famous Karolinska Institute. And the story of epigenetics actually began from Norrbotten, a small village in the Northern Alps in Sweden, where the population was just about a few hundred in 20-30 square kilometers. So this goes back to the, Dr. Lo Bygren was going back to the history of Norrbotten, and he saw that this area was totally geographically isolated because of its height, because there was snow. And in the 1800s, there were certain years when there was abundant harvest, and there were certain years when there was a famine. So during the years when there was abundant harvest, a lot of people indulged in gluttony. They ate away to glory. Dr. Lo Bygren observed that the kids and the grandkids of these people who ate and ate, their kids and grandkids died 32 years earlier than those kids whose grandparents did not eat so much when there was abundant harvest in Norrbotten. And this he attributed to genetic imprints of overeating which was left in the sperm and ova. And hence it was transmitted from one generation to another generation. Randy Jertle and Robert Watland of Duke University, he was an oncologist. He was working with mouse which had this agouti gene in it. Now this agouti gene in the mouse is responsible for giving it a yellow coat, a golden, nice, lovely yellow coat, and it predisposes the mouse to diabetes and obesity. What he did, he took two groups of pregnant mice and he exposed one to vitamin B12 and folic acid, and the other one was given normal diet. And when he found out that to the cohort to whom he had given vitamin B12 and folic acid rich diet, in them the coat was brown and they had normal weight and they did not have diabetes. So he hypothesized and proved that we all know that folic acid is a methyl donor. So he said that there was something from these vitamins which is actually down-regulating this agouti gene, and therefore the mice which were born out of this cohort in which B12 and folic acid was supplemented had normal weight and they had a brown coat because this agouti gene had got down-regulated. So this is the agouti gene, and you can see the mice which was born. This is brown and slender, and they did not have diabetes. Now the father of epigenetics was Conrad Waddington, and he was the first person to coin the term epigenetics. He's a famous biologist, paleontologist, anthropologist, and he a man of many skills. So what is epigenetics? It is the science which studies the changes in gene expression that are heritable, but do not involve changes in the DNA sequence. So that is the interesting part, that there is capability to change phenotypic outcomes which are heritable, but without changing the DNA sequence. And if we were to compare the epigenome and the genome, it's something like a genome is your hardware and the epigenome is the software which drives the hardware, which is your genome. So the science of epigenetics is also the one which can easily prove that when you look at two identical twins, they are born from the same zygote, and but one might develop schizophrenia later in life and the other one doesn't develop schizophrenia. Although the genetic component is same, it is due to the fact that they experience different environments as they grow. And this environmental change leads to certain outcomes like schizophrenia or bronchial asthma. So this very clearly, epigenetic elucidates that nurture drives nature. So the outcomes could be so different, just like Dr. Jekyll and Mr. Hyde. Now, coming back to the dynamics of epigenetics, if we were to look at the chromosome, this is the DNA, and this DNA is tightly wound around this protein discs, which are known as histones. As you can see over here, this is the histone disc and around which the DNA is very tightly coiled. And there is a histone tail which is jutting out. There are certain epigenetic factors. These epigenetic factors either get attached to the DNA, or it could get attached to the histone tail, which you can see jutting out. And due to this, there could be cancer. It could lead to autoimmune disease. It could lead to mental disorders. It could lead to diabetes. So epigenetics is now being all these autoimmune diseases, autism, schizophrenia, diabetes, everything is being attributed to environmental factors through epigenetic mechanisms. So how is the chromatin regulation? How does it happen? So if you look at the DNA, I said that it could happen at the histone level and at the DNA level. At the DNA level, there is cytosine methylation. As we know that there is ATCG, so there is cytosine methylation. And as the histone modification, there is much more variety. You could get acetylation, methylation, phosphorylation, and so on. Now, if you see, these are the different histones, H2A, 2B, 3, 4, and this is the genetic code. These letters basically denote all the different amino acids which make the proteins. And as you can see, there is flagging on this lysine. So this lysine has an acetyl molecule, so this is known as acetylation. Again, you could have here at this lysine molecule at the 4th position, you can see that there is acetylation as well as methylation. So this is how the histone modification occurs in the body. And there are other methods of modification as well. What are they? They are through the microRNAs. Now, microRNAs are actually non-coding RNAs. They do not code, and they are also responsible for post-transcriptional regulation of gene expression. And how do these mRNAs actually regulate gene expression? So these mRNAs actually nothing but they're small string of nucleotides, and they bind to the messenger RNA and cause their destruction by siRNA. siRNA is nothing but small interfering RNA, or they could destroy the mRNA by RISC complex, which is nothing but the RNA-induced silencing complex. So these are basically a string of miRNAs which could attach itself to different areas of the messenger RNA and destroy it, and thereby causing downregulation of particular genes. Now, methylation of messenger RNA is also known, and this occurs and it forms 6-methyladenosine or 5-methylcytidine. RNAs have their own methyl transferases and demethylases, and miRNAs are now being looked at as a very, very potential biomarker, and they have a lot of therapeutic potential in many, many diseases. So what if there is methylation? How does it eventually lead to expression or non-expression of a particular gene? So here you can see that, again, we are back to our histone discs around which the DNA is attached. And when the chromatin is in a euchromatin state, you see it's nicely spaced out. The histone discs are nicely spaced out. There is only one methyl attached over here. When this entire thing becomes hypermethylated, as you can see over here, there is 1, 2, 3, 4, 5 methyl molecules. When there is hypermethylation of the DNA over here, the whole thing has become very compacted. The histone discs have become compacted. And as a result, what happens? There is not enough space for transcription factors to come and bind. And hence, there is no production of messenger RNA, and the whole gene becomes downregulated. So basically, through the help of methylation, euchromatin is converted into heterochromatin. And when there is heterochromatin, the whole thing collapses. There is no attachment of transcription factors, and therefore the gene becomes downregulated. Now, let's look at the different mechanisms of how this happens. So this again is our histone disc. This is the histone tail. And if you can see over here, this there are certain writers and erasers. So this K is a lysine. K is the alphabet for the lysine. And over here you see this lysine methyl transferase. KMT is nothing but a lysine methyl transferase. This is transferring a methyl molecule, and therefore it has got the tail of this histone methylated. And how does this tag get erased? This tag will get erased when there's an eraser. It's like a lysine demethylase. So a lysine demethylase will remove. So eventually it's a tightrope walk. It's a balance between the methylases and the demethylases, which eventually determines the methylation level. So here again, if you look at this point over here down below, you see that there's this histone acetyl transferase. So this becomes a writer because what is it doing? It is transferring an acetyl molecule, and here you see that the histone tail has got acetylated. And histone deacetylases would be an eraser. It erases the histone, and then therefore the acetylation level would be eventually the ratio between histone acetylases and histone deacetylases. Then we have another interesting group of enzymes in our body which are known as the 10-11 translocases. Now these 10-11 translocases are present inherently in the body, which help in demethylation. They convert the methylated DNA into hydroxymethylcytosine. So therefore, this is a very important enzyme which governs the levels of methylation in the body, and they are known as 10-11 translocases. So basically what happens over here, with the help of epigenetics, there's a particular sequence in which a gene would get expressed or get downregulated, or